Hi, my name is Brianna McConnell, and my partners and I are here to discuss our research on the effects of hormone levels in dogs as a result of spaying and neutering them. First, to give you a brief medical introduction, spaying and neutering are surgical procedures in which an animal's reproductive organs are removed to prevent them from breeding. Now, neuter is a term that we use in reference to male animals, in which we remove the testes, and spaying is a term we use to refer to female animals, in which we remove the ovaries and sometimes the uterus. You should spay and neuter your dog for many reasons. First, for population control, as it decreases the number of potential stray animals in society, which can help reduce the chance of contracting zoonotic diseases and spreading those diseases throughout the community. Second, it prevents or diminishes behavioral problems such as roaming, humping, marking in the house, and aggression towards people and other animals. And finally, it's just been more beneficial to their health overall. Some of these health benefits includes that there will be no birthing complications for females, as they will no longer be able to produce offspring or have litters. So there will be no worry about the birth aftermath, as I like to call it, and any related problems associated with the birthing process. This can also lead to a decrease in appetite in the dog, which can decrease the chance of the dog becoming obese and further risk the chance of obesity related problems, such as high blood pressure, stuff like that. And finally, it can decrease hormone production within the dog, and that decreases the chance of cancer. Overall, this leads to a longer life expectancy for your animal. Now, to give you a brief introduction about cancer in dogs, cancer by definition is a disease in which abnormal cells divide without control and are able to invade other tissues via the blood and lymph systems. Now, there are many types of cancers in dogs, just as there are in humans. Some of these include bladder cancer, brain tumors, hemangiosarcomas, lymphoma, lung cancer, mast cell tumors, osteosarcoma, and skin cancer. However, for our research, we will be focusing on the reproductive cancers such as uterine cancer, ovarian cancer, testicular cancer, prostate cancer, and mammary carcinomas. Now, between males and females, there are different hormone concentrations and variations that can cause cancer. To start, we will look at hormone production that can cause cancer in females. Mammary tumors are caused by an excess of estrogen and progesterone. 50% of all tumors in female dogs are mammary tumors, and 45% of these tumors are malignant. Spaying your female before 2 years of age is the best method of prevention, and spaying her before the first estrous cycle is even more efficient and even more preventative. Uterine cancer is another type of cancer that females can get. It is caused by excess progesterone or outside sources of estrogen. It is usually benign and fairly uncommon, but it can cause abnormal or continuous estrous cycles and or painful urination. So it can cause outside problems that will affect the well-being of your pet. Finally, for females, we have ovarian cancer, which is caused, again, by excess estrogen and progesterone. Again, it is uncommon, but it's very serious when present. There are 3 types in dogs. There are epithelial tumors, germ cell tumors, and stromal tumors. The most common type of cancer in this spectrum are ovarian carcinomas. And this cancer is problematic because it is prone to metastasizing and can produce excess sexual hormones, which will essentially restart the cycle, and the excess of these sexual hormones will cause more cancers. So it's very problematic and should be controlled. And males, due to the differences in reproductive organs and anatomy, we have testicular cancer, which is caused by excess estrogen or estrogen-related chemicals and hormones and/or a decrease in testosterone. Is one of the more common diseases in intact male dogs, and the cells can metastasize to other parts of the body. That is less than 15% of cases, but when these cases are present, it is very serious for the animal. Dogs over the age of 6 have a higher risk for testicular cancer, and cryptorchid dogs, which a cryptorchid is one in which the testicles, one or both, have not descended, and they are 50% more susceptible to testicular cancer. If neither of the dog's testicles have descended, they have even a higher risk of getting testicular cancer. The other cancer seen in male intact dogs is prostate cancer, which again is caused by an excess estrogen or an excess or decreased amount of testosterone within the system. It's rare, but is almost always more common in intact dogs. It can lead to benign prostatic hypertrophy, or BPH, which is caused by an enlargement of the prostate, which blocks or restricts the flow of urine from the bladder, causing difficulty urinating. Again, this cancer is rare, but when it's present, it will create other issues for your pet and once again reduce the health of your pet and give your pet a painful sense of being. There are many ways to screen for cancer in your pets. The first is a biopsy, in which a small sample of tissue is taken surgically and then sent to a lab and examined and studied. This allows us to see if a lump or a mass on a dog is actually cancerous or if it's just benign, or it gives us a better indication of the severity of this lump. And if it does come back that the lump is cancerous and is malignant and it will pose a problem, then it can be removed by a simple procedure in a veterinary clinic. Next, we have a CBC, which is a complete blood count, and it is used to determine the red and white blood cell count within the dog's system. We use this because an increase in certain cells can indicate certain cancers or certain diseases present. For example, an increase in lymphocytes indicates that there is cancer present within the system. Next, we have a profile, which describes specific amounts of hormones within the dog's system. So we can use this and trace how much of a particular hormone is in a system. So for example, we can use a profile to determine how much estrogen is within a dog's system, or how much testosterone, or things of that nature. And this, we can compare this to other dogs or to a normal range that the dog should have, and we can determine if the dog is prone to cancer or prone to cancer-related issues. Finally, we have an autopsy, which of course can only be done once an animal is deceased. The benefit to doing an autopsy is if there is an unknown cause of a death in the animal, this will give a definitive diagnosis, and we'll be able to get a better view of the animal's entire body, and we won't be restricted by organs as we would with x-rays or ultrasounds. The importance of preventing cancer is probably obvious to most of us. It prevents the animal from experiencing severe pain and suffering. It prevents secondary illnesses and diseases. And honestly, it saves the owner money because cancer treatment is very expensive, and most owners are unable to afford it for their pet. So once again, it increases the overall life expectancy of your pet and leads to a very happy pet. Finally, I would like to introduce the breed that we will be using in our research. We will be using Golden Retrievers for various reasons. First of all, they are easy to train. They're very smart and very loyal, and because of this, they thrive in obedience and training classes. They're in sporting group, so they typically have a longer lifespan than some dogs, which the average is approximately 11 years. Um, they're family dogs and prefer to be with people, so they don't have um, any aggression towards people usually. They're very friendly, and they're easy to work with because they are so friendly. But the main reason that we decided to use Golden Retrievers is that they are one of the most susceptible dog breeds to getting cancer. 60% of Golden Retrievers will get cancer in their lifetime, which is double the 30% of other dogs that will get cancer within their lifetime. Hemangiosarcomas are the most common type of cancer in Golden Retrievers, and using a more cancer-prone breed, we will be more likely to get results for our experiment.
My name is Brianna Dorn, and I will be talking about the experimental portion of our project. For our experiment, we developed the hypothesis that dogs who are spayed or neutered will have an increased chance in having a non-reproductive cancer. We will be measuring this by using 4 litters of puppies, each litter containing 8 puppies, 4 female and 4 male. This is totaling 32 puppies, 16 males and 16 females. Of those puppies, 8 females will be spayed and 8 males will be neutered. We are using the Golden Retriever as our dog breed, as mentioned before, since it is the dog breed that is found to be the most susceptible to cancer. This in turn will amplify our results and either prove or disprove our hypothesis. We will be using our funding towards purchasing of the dogs, their food, medical expenses, and anything else needed for this experiment to proceed. As mentioned before, spay and neutering is where the veterinarian will remove reproductive organs, such as the testes or ovaries. In females, it's called an ovariohysterectomy, or spay. It is where the ovaries, fallopian tubes, and uterus is taken out. We take out the ovaries since it is the primary source that produces hormones like estrogen and progesterone, which we are going to see if influences the cause of cancer. In males, it is called an orchiectomy, or a neuter, which is the removal of the testes. We remove the testes in order to take away the primary source of producing hormones like testosterone, which again we will be seeing if it influences any cause of these cancers. In order to prove that dogs are getting or not getting cancer directly as a result of the spay and neuter, we have to eliminate other outside variables such as the environment. Doing this, we will be keeping their food, exercise, as well as many other variables constant. We will do this by keeping them on the science diet, which is approved by a variety of veterinarians. Also, we will be having their exercise at least 1 hour a day. We will also keep them exposed to the same air, water, bedding, attention, and overall living quarters. We will do this by keeping them in the same laboratory, having them play in the same yard, as well as the same room. In order to prove or disprove our hypothesis, we are going to be taking blood work from each of the dogs. This blood work will entail in a CBC, or complete blood count, as previously stated, as well as a complete profile. A CBC would tell us a variety of things, such as a red blood cell count, which includes measuring the hemoglobin and hematocrit, or PVC, to check if they are anemic or dehydrated. We will be primarily focusing on the white blood cells, more specifically the lymphocytes, which, if elevated, could indicate cancer. We will be collecting these blood samples yearly until they reach the age of 6, and then we will collect blood samples twice a year. We do this because at the age of 6, dogs are considered seniors, and as the dog increases age, it also increases in health problems. This experiment will conclude after all of the subjects are deceased. Due to varying health, this could average to around 12 years. Once every dog has died, a necropsy will take place to see if there are any signs of cancer or any other health problems that the blood work may or may not have picked up on. Also, any other signs or reasons for their death will have to be concluded as a result.
Hi, my name is Samantha Minard, and I will be discussing the results and conclusions section of our research proposal and presentation. So what we had found during this whole research proposal is that in neutered males and spayed females, they have a much higher risk of getting a generalized type of cancers. The highest percentage we had found was lymphosarcomas, hemangiosarcomas, and mast cell tumors. This being the reason because when animals get spayed and neutered, there's no longer parts that can make these hormones. So such things as gonadal hormones, gonadotropin hormones, and follicle stimulating hormones are basically cut off. And when this happens, they still have the receptor sites in the animal. So such things as cancer cells will now more easily attach to these receptor cells, which will allow them to grow a lot easier and make it a lot more common when these common hormones are not making their daily, growing, dying, and reproducing. So in this experiment, we found that the generalized cancers in these spayed females and neutered males were increased by 3 to 4 times when you consider them having one or more cancers. So more specifically, neutered males at having a rate or having the cancers with one or more, these were found to have 15 to 17%. As in the spayed females, they had a rate of at least one or more cancers at 8 to 14%. So in contrast, the incidence of cancers in intact females were around just 3%, and the rate of intact males was just around 4%. So you can see how much large of a difference the spayed females and neutered males had for these one or more cancers compared to the intact animals. So here we can look at this chart. It better and more easily explains the numbers we had found with these cancers. If you look at the left column, LSA stands for lymphosarcoma. The middle column, MCT, stands for mast cell tumors. And then you have HSA, which stands for hemangiosarcomas. You can see that the male intact, the ratio for that, and then the female intact. You can see the females that were spayed and the males that were neutered, they have a much higher rate or risk of getting these cancers. You can see, for instance, the males from 6 to 11 months were at a rate of 11.48%, and then you have the females at 6 to 11 months were at a rate of 10.98. So in this experiment, we had also looked at some other types of problems that arose from getting your animal spayed or neutered, and what we found was a large amount or an increased amount of joint disorders. These joint disorders were found for an animal having at least one or more. The neutered males were found to have an increased rate of 12.5%, and the spayed females, for the occurrence of having one or more joint disorders, was about 20%. And in contrast again, for the intact males, they had a rate of getting one or more joint disorders at 5%, when with the rate of the intact female, her chances of getting one or more joint disorders was just set as 5% as well. And again, you can see this chart. The left column stands for hip dysplasia, middle column stands for cranial cruciate ligament tears, and the right column, ED, stands for elbow dysplasia, Those were the 3 most common joint disorders we found. You can see the female intact and the male intact. You can see, once again, the rate. The female at less than 6 months that was spayed had a lot higher rate than the intact female, as well as the male, who had a much higher rate at less than 6 months of being neutered than the intact male. So, as with any research proposal and presentation, you can have sources of error. And even more so because this experiment takes years and years to actually find and test and finish your experiment. With these living animals, they all have different owners. They all eat different food. These animals as well can come into contact with different diseases. We can look some of this set of litters, they can have pre-existing conditions from the parents or anywhere in the line. The environment can also have a big effect on these animals, considering if they might be in a more stressful environment versus an animal that is in not a stressful environment. You can also have other pets in the house that can give them different diseases or just have them grow up a different way and eventually die. So in conclusion, we had found that neutering or spaying your animals increases their risk of getting a generalized cancer. But in another sense, not spaying or neutering your animals can increase their chance of any reproductive cancers. So this kind of created a problem for us. You can either not neuter or spay your animal with these pros and cons, or you can spay and neuter your animals with these pros and cons. So we had collectively decided that ultimately this decision is going to be up to the veterinarian and client together. You'll each have to check certain pros and cons and different scenarios. Ultimately, they're going to have an increased risk of cancer no matter what way you take. So it's honestly up to the veterinarian and client to what they think is best. As well as we had found that you can increase or broaden this type of research proposal by eventually testing it on different breeds of dogs, as well as different species of domestic animals such as cats. So overall, to conclude everything, animals that are spayed and neutered have an increased risk of generalized cancer such as lymphosarcoma, hemangiosarcoma, and mast cell tumors, but animals that are not spayed and neutered have an increased risk of reproductive cancers. And to conclude, here are our sources.